In this video, we learn about the process of gene transcription. The instructions for all the proteins that are made inside a cell come from the genome or the DNA of the organism. According to the central dogma of molecular biology, these instructions are first used to build mRNA molecules, which are synthesized in the nucleus and transported out of the nucleus into the cytoplasm, delivering these instructions to the site of protein synthesis where proteins are built. So the first step in converting genetic information into proteins is through transcription. The enzyme RNA polymerase performs this synthesis by transcribing only one strand of DNA called the template strand. The other strand is called the non-template or coding strand. The sequence of this strand matches the sequence of the mRNA, except that RNA has uracil in place of thymine. RNA polymerase uses the information in DNA to build RNA molecules in the 5' to 3' direction. This enzyme catalyzes the formation of phosphodiester bonds between nucleoside triphosphates. Like DNA polymerases, an RNA polymerase also performs template-directed synthesis in the 5' to 3' direction. But unlike DNA polymerases, RNA polymerases don't require a primer to begin transcription. This is also the reason why primase, which is a kind of RNA polymerase, is able to synthesize an RNA primer during DNA replication. Bacteria have only one RNA polymerase. Eukaryotes have 3 distinct types of RNA polymerases, 1, 2, and 3. So how does transcription begin? The first stage of transcription is called initiation. To begin transcription, RNA polymerase needs some help. This help is provided by a protein subunit called sigma that first binds to polymerase, forming a holoenzyme. A holoenzyme is basically a protein complex made up of a core enzyme and additional required proteins that together serve a particular function, which in this case is beginning the process of transcription. Prokaryotic RNA polymerase is also a holoenzyme that is made up of the core enzyme and a sigma subunit. The sigma subunit acts as a regulatory factor. It guides the RNA polymerase to specific sequences within the DNA template called promoter sequences. Bacterial promoters have some key features. They're approximately 40 to 50 base pairs in length and consist of key regions, one of which is known as the -10 box. This is because it is present 10 bases upstream of the transcription start site, which is indicated by the +1 sign. This site is where the first ribonucleoside triphosphate will be added. The -10 box typically consists of the sequence TA-TAAT. There is another key region called the -35 box, named because it is found approximately 35 bases upstream of the transcription start site. This consists of the sequence TTGACA. All bacterial promoters have these 2 key regions that help the sigma subunit recognize the promoter and bind to it. The rest of the promoter sequence can vary. After the sigma subunit binds the promoter, it helps in opening the DNA double helix such that the template strand is threaded through the RNA polymerase's active site. Subsequently, an incoming ribonucleoside triphosphate, or NTP, pairs with a complementary base on the DNA template strand, and this is how RNA polymerization begins, at the +1 site. Once this process begins, sigma dissociates from the core enzyme, completing the initiation phase of transcription. RNA polymerase can then move in the 5' to 3' direction along the template DNA strand. The next phase in transcription is the elongation phase, during which RNA polymerase moves along the DNA template, synthesizing RNA in the 5' to 3' direction. Finally, transcription ends with the termination phase, during which RNA polymerase synthesizes RNA that forms a hairpin loop structure, signaling the end of transcription. At this point, RNA polymerase dissociates from the DNA and transcription is complete. The newly synthesized mRNA molecule is now ready to undergo further processing. Eukaryotic promoters are similar to bacterial promoters, but they're much more diverse and complex because eukaryotes typically have more genes that are separated along the length of chromosomes. Many of the eukaryotic promoters include a unique sequence called the Tata box, which is centered about 30 base pairs upstream of the transcription start site or the +1 site. As with bacteria, the RNA polymerase in eukaryotes does not directly bind to the promoter. A group of proteins called basal transcription factors bind to the DNA promoter and help RNA polymerase initiate transcription. These basal transcription factors perform a similar function to bacterial sigma proteins, but instead of just one subunit like the sigma subunit, there are multiple proteins that come together to perform that function. Let's look at a summary of the process of transcription. Transcription takes place in the nucleus during interphase of the cell cycle. If you zoom in, a region of DNA is unwound and a strand of RNA complementary to one strand of the DNA is produced by the addition of RNA nucleotides. So this strand in DNA serves as the template strand, which directs the synthesis of the new RNA strand. The newly synthesized mRNA strand can now exit the nucleus into the cytoplasm. In bacteria, the information in DNA is converted to mRNA directly. Note here that bacteria don't have a nucleus, and so as soon as the mRNA molecules are synthesized, they can be used to make new proteins inside the cell. In eukaryotes, before the information in mRNA can be used to synthesize proteins, the immature primary transcript or pre-mRNA needs to undergo certain processing steps in order to prepare for translation. The pre-mRNA molecules need to be processed because the protein coding regions of eukaryotic genes are interrupted by several non-coding regions that need to be removed before the mRNA can be translated to form proteins. The coding regions of eukaryotic genes are called exons. These exons will come together to form the final mRNA product. The non-coding regions or the intervening sequences are called the introns that don't end up in the final mRNA. So eukaryotic genes are in fact much larger than their actual corresponding mature mRNA. The introns within eukaryotic RNA transcripts are removed by a process called splicing. As we can see in this image, there are a lot of extra sequences within the primary RNA transcript called introns that need to be removed. This removal of the unnecessary sequences is catalyzed by a complex known as the spliceosome. The final mRNA molecule ends up being much shorter than the original sequence contained in the template DNA. In addition to splicing, there are certain other modifications made to the RNA transcript before it is completely ready for the process of making proteins. These modifications include the addition of a 5' cap that serves as a recognition signal for the translation machinery, and a poly A tail, which adds stability to the mRNA molecule, protecting it from degradation. This extends the life of the mRNA molecule. This concludes our section on gene transcription. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.